Hello friends, I think you must have listened to the mitosis lecture carefully. Now it is a time for meiosis. Meiosis, many times students they think that this particular topic meiosis or any cell division type that is too difficult to understand. Let me tell you once again, be relaxed. Whatever the things are happening, they are happening in a cell, single cell. There are so many millions, trillion cells are present in a body. Whenever the things are taking place in either mitosis or meiosis, they are just steps. It's a series. It's a what I can say. It's a sequence. Single cell is available. Certain processes occur in the cell, and that cell divides into two. So this is mitosis. Similarly, there is certain category of cell, that is sex cells, or the cells which are responsible for continuation of life, that is reproduction. These cells they involve themselves in cell division, but a little different type of cell division they carry out. So this particular cell division is nothing but the meiosis. In mitosis, you get one cell divided into two, that is product is two cells, and the product of meiosis is four cells. I think this much basic information you have learnt in your school days. Okay? Now, let us go beyond the basic knowledge. Let us think about the ploidy level. I know many of you are finding these particular statements, ploidy, diploid, haploid, they must be finding this particular knowledge very easy, but there are many students, including myself, those who feel this particular meiosis part little tricky. It is quite natural. Even today, when we go through certain books, there are so many reference books which are here at Learning Center. We always refer them. Even I don't understand many things, so I can ask my doubts to Acharya sir or Kaustubh sir. So I can get my doubts cleared. Same thing can be there with your case. So definitely you can come across many difficulties. So you keep on reading. Once you keep on reading, you will feel comfortable, you will feel acclimatized with the topic, and then you can ask us doubts. Okay? So this is the basic framework of learning. So let us think about the meiosis once again. It's a special type of cell division you can call. Okay? This special type of cell division occurs in sex cells, that is the cells which are responsible for reproduction. Okay, consider in case of humans. Let us begin with the humans. In case of humans, sperms and egg cells are produced. Sperms, they are haploid. Deliberately I am using some technical words. Haploid sperms are produced. Egg cells are also haploid. They are produced by meiosis. Let us think about the plants. When we talk about sexual reproduction in angiosperms, that time we always think about the production of pollen grains. Pollen grains, as a result of pollination, they fall on a stigma and they produce pollen tube, and pollen tube passes through the stylar canal and reaches up to the egg, and further the fertilization and zygote and embryo and seed setting, everything occurs. Let me tell you in the beginning, this particular meiosis process always deals with the sex cells. It always deals with the sex cells. Product of meiosis is 4 haploid cells. Means there should be a diploid cell in the beginning. Unless and until the basic parent cell is diploid, you cannot get 4 haploid cells. So you can say, in other words, you can say product of meiosis is 4 haploid cells. Always remember, product of meiosis is 4 haploid cells. Let us read through the, let us go through the certain technical issues and let us read the things together. Meiosis, fantastic mechanism. It is a well set program by the nature. We cannot say any god or any superpower here. So simply say it's a nature, right? As a meiosis, if you think about the process, it's a simple process. If you think about its importance, then it will be, I can say, it's a tremendous thing. In what way? Let me tell you. Suppose meiosis is not there. We people are getting double by binary fission or by mitosis. Suppose the person who is looking similar to me, so he is present here after a certain age or a certain period. So by mitosis, I must be dividing. So definitely population will increase. But whatever the things are getting produced from a single animal, say me. So whatever the product is available, whatever the daughter cells, daughter body is available, it will be similar to that of parent body. Whatever the requirement is there for food, space, shelter, reproduction will be the same type. So my population will be huge with same genetic makeup. And ultimately, what will happen? Suppose if any disease spreads there in my community, in my area, in my territory, that time, if I catch that infection easily, whatever the population is there along with me, who is having genetic makeup exactly similar to my genetic makeup, then definitely entire population will get that infection. It will be a monoculture type of thing. So there should be some mixing of genes. Let me tell you, mixing of genes always gives a better result, best result I can say. When the Sir Mendel were working, he was working along with his co-workers, so the team when it was working. So he came to know about, he struck something that definitely there must be some segregation of factors. So law of segregation is there, you must have studied, or if you are not remembering, don't worry, you are going to learn that particular topic once again in standard 12. Okay, so don't worry about it. So this is how the segregation of genes is required. So whenever there is segregation of genes, once again there is a great chance of recombination, and when the dominant type of genes are available, they express themselves. So whenever the crossing over is possible, the recombination is taking place, that time there are two set of chromosomes, different set of chromosomes you can consider. So best genes from this particular one and best genes from this one, so they can be clubbed together, and this is how the new product, new individual can be formed with the new characteristic features. So it is possible. So this is how meiosis, it has a great importance in evolution. Let us think about certain key issues. Interphase. Interphase is another, what I can say, most important step in meiosis 2. We have seen this particular concept of interphase in mitosis. Similarly, interphase is available in meiosis. So there should be some preparation, there should be some DNA replication. Okay, so unless and until the DNA is replicated, the cell cannot move further for the various sequential processes. Okay, so this entire meiosis part we can split into two phases, meiosis 1 and meiosis 2. Meiosis 1 is a reduction division. Why is it called as reduction division? You can read here. Number of chromosomes, they get reduced to half. When we say meiosis, entire meiosis is a reduction division, that time we need to focus on meiosis 1, where exactly the chromosome number get halved. So this is, suppose here X chromosomes are available, or other any alphabet you can take, say Z chromosomes are available, and if meiosis occurs, or meiosis 1, so this becomes half, X by, or what the letter was, Z by 2, so this will be the number, okay. Chiasmata is another technical term. Chiasmata means you can say a pairing. Pairing can be there. Let me show you this typical process. Suppose these are the chromosomes, and if they are getting attached to each other, and if they are sharing some chemicals, actually chromosome is nothing but the it's a compact DNA. If some piece, suppose this much piece is getting replaced from here, and if it is getting fixed here, and this piece is getting exchanged with this piece, so this is how the crossing over occurs. So this is chiasmata. No new terms will be there, only technical terms will be added. Whatever the process is there, process remains same. Exchange, crossing over, chiasmata formation, nodule formation, whatever the things are there, you need not to bother about it. Okay? So this is meiosis 1, where the reduction division occurs. So it can be called as heterotypic type of division. Okay? This particular word, heterotypic and homotypic, this particular these words they have used in state board book. NCERT book, they may not show such type of technical terms, but let me tell you in the beginning itself, for this particular chapter there are so many technical terms that you need to remember, but in the beginning simply go slow, do not make any hurry, and do not get worried if you don't understand anything, even after listening to this lecture at least 20 times. हे समजा लक्षात आलं नाही, अगदी 20, 25, 30 वेळा ऐकल्यानंतर सुद्धा काळजी करण्याचं कारण नाही, याचा अर्थ तुम्हाला निश्चितपणे चांगलं समजणार आहे. पण याचाच अर्थ एक असा आहे, हा चॅप्टर व्यवस्थित कळाल्याशिवाय तुम्हाला पुढचं जेनेटिक्स प्रकार जो आहे तो कळणार नाहीये. कारण जेनेटिक्समध्ये जीन्स आले, जीन्स म्हणजे डीएनएचे तुकडे आले असं समजा, आणि डीएनए कसा असतो, कॉम्पॅक्टली पॅक केलेला क्रोमोझोम, बरोबर आहे. तुम्हाला बेसिक गोष्टी येत आहेत, आपण अनकॉइलिंग करत चालू, क्रोमोझोम अनकॉइलिंग केलं, मोठा डीएनए फ्रॅगमेंट, मग त्यातले जे काही पीसेस आहेत ते जीन्स येतात, मग त्याच्यावरती त्यांचे ते कुठलेही कॅरेक्टर असेल, सपोज दॅट पर्टिक्युलर जीन रिप्रेझेंट्स अ कॅरेक्टर फॉर नोज, फॉर इयर, फॉर आय, फॉर आयलिड शेप, फॉर हेअर्स, हेअर कलर, किती प्रकारचे कॅरेक्टर्स असतील ते सगळे त्या डीएनए मध्ये असतात, म्हणजेच त्या क्रोमोझोम मध्ये असतात. आता हे सगळं जेनेटिक्स आहे आणि हे मिओसिस आहे, याच्यात क्रॉसिंग ओव्हर होतं, फर्टिलायझेशनच्या वेळेस जे काही एक्सचेंज होत असतील या सगळ्या गोष्टी इथे होतात. त्याच्या आधी ते हॅप्लॉइड सेल्स तयार व्हायला पाहिजेत, ती सगळी प्रोसेस आपण इथे शिकतोय. लक्षात येत आहे, हा बेस आहे पुढच्या ह्युमन रिप्रोडक्शन चॅप्टरचा, जेनेटिक्स चॅप्टरचा, बायोटेक्नॉलॉजीचा. कारण फक्त ह्युमन्स मध्ये आपण जीन शिकणार नाहीये. तुमच्यातले अनेक जण बायोटेक्नॉलॉजीमध्ये म्हणतात की आम्हाला करिअर करायचं आहे. ठीक आहे, एखादी सीड कंपनी आहे, XYZ सीड कंपनी आहे, त्याच्यामध्ये जेनेटिक प्रोग्रामर लागत असतील आणि तिथे जर का तुम्हाला त्यांची बायोटेक डिव्हिजन तुम्हाला सांभाळायला दिली, नवीन प्रोडक्शन काही करायचं आहे, तुम्हाला जेनेटिक रिकॉम्बिनेशन्स करायचे आहेत, जेनेटिक इंजिनिअरिंग, कटिंग जॉईनिंग, बरोबर, हे सगळं करताना तुम्हाला हाच बेस लागणार आहे. त्यावेळेस तुम्हाला वाटेल अरे आपल्याला त्यावेळेस ते जमलं नव्हतं, आता किती इझी आहे. तसंच असतं. एकदा टॉपिक तुम्ही शिकायला सुरुवात केला तो फक्त वाचत राहा. ऑटोमॅटिकली यू विल गेट यूज टू दॅट पर्टिक्युलर सेट ऑफ टर्म्स. And then you will be very fluent with all the concepts. You will be very fluent with all the technical issues. Okay. Now, think about the meiosis 2. Meiosis 2 is homotypic or homotypic, or you can say it's an equational division. Meiosis 2 is a simple mitosis. You can say mitosis. You have learnt well. Meiosis 1 is little tricky, not much, and meiosis 2 is similar to that of mitosis only. So hence it is called as equational division. One cell divides into two. As a product of entire meiosis process, there are 4 haploid cells are formed from single diploid cell. आता पहिल्या स्टेज मध्ये रिडक्शन झालं, दुसऱ्या स्टेज मध्ये इक्वेशनल डिव्हिजन झालं. त्यामुळे इथे काय झालं, 2 सेल्स तयार झाल्या. पुन्हा तिची प्रत्येक एक सेल होती, त्यामध्ये मायटोसिस झालं आणि त्याच्यामधून 4 सेल्स तयार झाल्या. हा एवढा बेसिक सरळ हिशोब आहे. तुम्ही म्हणाले तुम्ही काही सांगतात आम्ही वाचत होतो, आमच्या 8, 9, 10th मध्ये आम्ही वाचलं पण काही लक्षात नाही आलं. बरोबर आहे, कसं काय येईल लक्षात? ती गोष्ट तुम्ही एकदाच वाचली. त्यावेळेस सायन्स हा फक्त एक सब्जेक्ट होता. आता सायन्स ही ब्रांच झालेली आहे. लॉजिक तुमचं वाढायला पाहिजे. तुम्ही बायोलॉजी हा एक वेगळा विषय म्हणून घेतलेला आहे. त्याच्यातले तुम्हाला टेक्निकल डिटेल्स जास्त येणार आहेत आणि ज्यावेळेस हे टेक्निकल डिटेल्स येत आहेत त्यावेळेस तुमची थोडी तयारी जास्त पाहिजे की नाही हे थोडं अवघड आहे पण मी करू शकतो, एवढी तुमची तयारी असेल. हे काय याच्यापेक्षा अजून किती मोठं तुम्हाला शिकायचं, हे तर काहीच नाही, हे किरकोळ आहे. आणि हे एकदा झालं, हे समजलं, मग समजून घ्या तुम्हाला पुढचंही सगळं जमलं. कारण हा बेस आहे, सगळ्याचा बेस सेल बायोलॉजी आणि सेल डिव्हिजन. तुमच्या लक्षात येत आहे सगळं. त्यामुळे मला वाटतंय की हा एक बऱ्यापैकी तुम्हाला अंदाज आलेला प्रकार आहे. सो हियर आय एम रेफरिंग दिस पर्टिक्युलर मटेरियल and I think page number 22 to 26 is a set of 5 pages, or in your case it must be printed on both sides. So 2.5 pages set it will be there. So this particular set is for cell division chapter, and I think the diagram sheet which is present here, yes. So there are 2 major pages. First page is all about mitosis. This particular part we have done, and this second part is about meiosis. So this is how we are going to learn each and everything. Now I'm going to take a pause for a few seconds, and then we are going to start with the actual concept. So I think you should get ready with your notes, diagram sheets, entrance notebook, practice notebook if required, and some color paints and some other writing material or the material which you require. Along with this, you should have your NCERT and board book. So whenever I ask you to refer page number so and so of your board book, refer so and so page of your NCERT, that time you should refer it. From time to time, I am going to take a small pause, small break, a break of say a few seconds. That time you should refer that particular material. So this will help you to understand what matter is required to learn. You need not to mug up each and everything. There are so many details which are given there in your board book. There are so many diagrams are there in your board book. Definitely we are going to tell you that this particular diagram you should strike it out. You should not refer it. Simply refer the diagram sheet which is provided by Learning Center. So this will help you a lot. This is a really simplified study material which is available. Along with this particular set of instructions, let me tell you once again, do not refer any extra material. It will waste your time, it will create chaos, it will create confusion, and you will not be able to focus on the concept which is required there, which is expected from you people. Okay, so get ready. Let us continue with a small pause. So I think let me start with my mic. Yes, now I can speak better. Let us start with the actual concept of chromosome once again. Why we require brushing up the knowledge for the chromosome? Because whenever we think about crossing over, we are going to think about chromosomes, we are going to think about DNA molecule, we are going to think about genes. When we think about, there is a combination of a new combination is available with the product, that is a baby or a offspring. So that time we always think about recombination, and recombination comes through the chromosomes. Let us draw a chromosome with two chromatids with some different color. I can show centromere. Okay, now this particular chromosome, in case of humans, there are 23 pairs of chromosomes, 22 plus 1 pair for 6 chromosomes. Always remember, when they are present in pairs, for our convenience I'm drawing in a simple form, when they are present in pairs, so one pair come from father and one pair come from mother. So this particular set is called as paternal and maternal set. So this particular set comes from our parents. Okay, understood? So similar to this is pair 1. So this is how total 23 pairs are present in humans. Whenever we will focus the camera on the board, you will find total 4 pairs of chromosomes. So that is just for our convenience. And let me tell you in other words, we are not learning everything about humans. We are generally learning the meiosis process. We are generally learning the biology, and biology can be with anything. It can be with some algae, it can be with giant human or elephant or butterfly, giraffe, monkey, anything. Okay, so that's why I have explained this particular concept in the beginning. Now let us consider a typical chromosome and the concept of homologous chromosome. Suppose this is a chromosome, and let us think about the X, Y and Z genes. Suppose here, somewhere here, the X gene is present. Somewhere here, Y gene is present. Somewhere here, the Z gene is present. This particular representation was used by Kausupsar while discussing these things. So I am using the same thing. Now, think about this second half, this particular another arm, you can say. This particular location also represents the same gene, that is X. This location also represents the same gene, that is Y. The same location represents the same gene, Z. For our convenience, we are considering this X, Y and Z type of genes. Now, these two, what I can say, arms are called as the sister arms. Okay. Sister arms. Let us try to remember the new word or the proper word for this. sister chromatids. So these are the sister arms or sister chromatids, you can say, on which the X, Y, Z genes are present on the same locations. Okay. Now think about the recombination. Recombination will not occur here. This particular X and X will not get mixed with each other. Suppose a chromosome, another chromosome with the same properties appear here. Once again, let us show the centromere with another color and let us represent the genes. X, Y, and somewhere here Z. Similarly, here X, Y, and Z. Okay. Have you understood this? This particular chromosome number 1, this is chromosome number 2. These chromosomes are different, but they are homologous. What do you mean by homologous? When you think about this gene X, gene Y, gene Z, same gene which is representing the same property of individual, say a gene for nose, gene for ear, and gene for hair color. Same type of genes are present on the another chromosome. Okay, this is another chromosome, but this is homologous chromosome. Same dimensions, same shape, same gene sequence, everything is same. So the gene responsible for X property will be on the same loci of the another chromosome. Okay. Now let us think about the recombination. Okay. What we are going to learn? Recombination. How recombination occurs? There should be some barter, exchange. There should be some exchange between two chromosomes. You have learnt well about this particular concept in your cell biology nucleus part. This particular chromatid, let us name them, say chromatid 1, chromatid 2, chromatid 3, and chromatid 4. Now the principle is that this particular chromosome should exchange the genes with the another chromosome, not with the same arm, not with the sister arm. Okay, so the combination can be like this. So I'll write the word possible recombinations, not combinations. Possible recombinations. This C1 can exchange material with C3 and C4. Okay? C3 and C4. Now you can tell me about C2. C2 can exchange material with C3 and C4. You got it? Now let us think about the another combination. Think about C3. C3 can exchange its material with C1 and C2. Can it show any recombination with C4? No, it is not possible. So it's a sister chromatid. both of them, they are showing the same structure. These are homologous, but still they can exchange the genes or piece of DNA, you can say. If you uncoil this is compact form. If you uncoil this, a long thread will be there. On that thread, particular sequence, you need to pick from this piece and you need to join there. From that particular part, you need to pick that particular part and fix it here. So this is how recombination occurs. Similarly, C4. C4 can exchange its material with C1 and C2. It cannot exchange its gene with C3. Once again, C1 and C2. So this is very basic concept. We are going through this concept very quickly just because we need to understand how various stages are there in actual meiosis type of cell division. Okay, so I think with this particular basic knowledge we can shift to the actual board. Yes. Let us think about the various stages. So, sir, is it possible for we people to just have a look through all these stages?
Yes, sure.
So, these are the stages. Don't worry about their names. Say A, B, C, D, and E. These are the stages. How these stages appear, how they are there on the board. What is this leptotin and what is this diplotin? Everything we are going to learn. Don't worry about it. Now, let us learn the topic thoroughly. Let us think about the page number 24 of your notes. So, are you ready with your notes? Yes or no? No? Yes, you get your notes. Pause the video. Get your notes. Get your diagram sheets. Be ready. Be alert. Okay? On this particular page number 24, on this particular page 24, you can find everything. The mitosis part we are going to learn later, but this is meiosis, meiosis 1 and various stages, everything is there. Page number 24 we are referring. So you should be very sincere and serious about the topics and the methodology we are using here and we are sharing with you. So meiosis. Meiosis is divided into two phases, that is meiosis 1 and meiosis 2. Meiosis 1, which is reduction division, and it is subdivided into, once again, 4 substages, that is Prophase 1, Metaphase 1, Anaphase 1 and Telophase 1. As soon as the cell ends with the Telophase 1, meiosis 2 starts. Once again, meiosis 2 is divided into, once again, 4 substages. Prophase, can you read it there? Yeah, you should read. Prophase 1, Prophase 2, then Metaphase 2, Anaphase 2 and Telophase 2. Meiosis 1 has 4 stages, meiosis 2 has 4 stages, total 8 stages, and the gap which is present between these two phases, meiosis 1 and meiosis 2, is called as interphase. Interphase, you can see here, it's a short period actually, or you can not interphase, my mistake, interkinesis. Interkinesis is a transition period. What is transition period? When particular cell, it undergoes certain stages of operations. When it reaches to the telophase 1, that is end of meiosis 1, there is a, I cannot say it's a lag actually, but there should be some transition from phase 1 to phase 2. So it's a short phase actually. But yes, definitely it appears there. So this particular transition period between meiosis 1 and meiosis 2 is called as interkinesis. It's a short phase. Okay. So what we have done with the concepts of meiosis, necessity of meiosis, stages of meiosis, that is meiosis and meiosis 2, meiosis 1 and meiosis 2. Meiosis 1 divisible into 4 subphases, that is Prophase 1, Metaphase 1, Anaphase 1, Telophase 1. Similarly, on the other hand, interkinesis, that is gap phase or short bridging phase has occurred. Now the cell has entered into the second stage of meiosis, that is meiosis 2. So it has 4 substages once more, that is Prophase 2, Metaphase 2, Anaphase 2, Telophase 2. Prophase, Metaphase, Anaphase, Telophase. This sequence you need to remember. You need to put simply 1, 1, 1, 1. Then in meiosis 2, once again put 2, 2, 2, 2. That is simple, very simple. Okay. Now, once again, focus on your notes. Always remember, whenever we ask you to refer your notes, we have started with the chapter, go through the entire set of notes, that particular chapter. For that particular chapter, you should read each and everything. So we'll come to know about the various key concepts. At least you'll get familiar with the spellings of the words. Okay, so meiosis 1. Meiosis 1, it starts with the prophase 1. Here, this particular part is little lengthy. Actually, that part which we are going to learn is lengthy, but actually in the cell itself, the prophase 1 is quite lengthy. Okay, this prophase 1, it shows 5 stages. What are those stages? Stage 1, stage 2, stage 3, stage 4, stage 5. तुम्ही म्हणाल की आम्हाला ही काय नावं तुम्ही मगाशी म्हणलात, लिप्टोटीन आणि डिप्लोटीन आणि पॅचिटीन सगळं दिसतंय आम्हाला, आणि तुम्ही आता तर सांगितलं स्टेज 1, 2, 3, 4, 5. ठीक आहे, मला हे नाही लक्षात ठेवायचं. बरं, मला ABCDच म्हणायचं आहे तरी चालेल. स्टेज A, स्टेज B, स्टेज C, स्टेज D, स्टेज E. मला एवढंच म्हणायचं आहे. किंवा हेही सोडून द्या, मला फक्त फेज 1, फेज 2, फेज 3, फेज 4, फेज 5. प्रोफेज 1च्या 5 फेजेस तेवढ्या मी लक्षात ठेवल्या. काय होतं ती प्रॉपर्टी लक्षात ठेवली. कुठे क्रॉसिंग ओव्हर झालं ते बघितलं, कुठे जीन सेक्शन झाले तेवढे मी लक्षात ठेवले. बाकी तुम्ही नावं काही द्या ना, त्याला काहीच फरक पडत नाही. ठीक आहे. सो डू नॉट वरी अबाउट दीज वर्ड्स. These words have Greek origin and you need not to worry about them. Whenever you feel comfortable that time, even after 12th, if you feel comfortable that time, you go and search for these words. हे कुठलेही शब्द तुम्हाला ॲक्च्युली आता लगेच त्रास देणारे नाही आहेत. राहिले लक्षात ठीक राहतात सगळ्यांच्या, आणि नाही राहिले लक्षात तरी काही फरक पडत नाही. बऱ्याच जणांचे विसरतात, माझे विसरतात. मला सुद्धा अरे हां लेप्टोटीन आहे का, बरं होतं ना, असं होऊ शकतं. एवढ्या सगळ्या गोष्टी आपल्या डोक्यात असतात, त्याच्यात हे एक लेप्टोटीन थोडं विसरलं म्हणून काय झालं. इव्हेंट माझ्या लक्षात आहे, प्रोफेज 1 आहे, सगळी प्रिपरेशन चालू आहे, हे मी काय बॅक ऑफ द माइंड लक्षात ठेवेन. मी प्रोफेज ज्यावेळेस मायटॉसिसला शिकलो त्यावेळेस मला माहिती होतं की हे सगळं मटेरियल इकडे तिकडे स्कॅटर्ड होतं, आता ते कॉम्पॅक्ट होत आहे, लूज थ्रेड्स होते, कॉम्पॅक्ट होत आहेत. प्रॉमिनंटली कुठल्या क्रोमोझोम दिसण्याची शक्यता नव्हती, ते दिसणार आहे आता. ह्या सगळ्या बेसिक कन्सेप्ट्सवरती मी पुढे जातोय. त्यामुळे व्हेन आय स्टार्ट विथ ॲक्च्युली फेज 1 और A, that time I know something is going to be there, something is getting ready for next time. Okay, let us go through the actual words now. आपल्याला फक्त आता हे बघायचं, हे सोडून घ्या, हे आपल्याकडे नाहीच आहे, फक्त एवढं बघायचं आपण. In case of humans, there are 23 pairs of chromosomes. इथे किती दाखवल्यात मी? कुठले एक कलर घ्या तुम्ही. 1, 2, 3, 4. आपल्या कन्व्हिनियन्ससाठी 4. तुम्हाला वाटलं 2च घ्या, काही फरक पडत नाही. एखाद्याला वाटलं 3 घ्यावे, 3 घ्या. शेवटी आपल्याला काय कन्सेप्ट कशामुळे कळते त्यासाठी या सगळ्या गोष्टी आहेत. लेट अस थिंक अबाउट द ॲक्च्युअल न्यूक्लियस नाव. हो, सो इथे हा जो आहे प्रकार, हा काय दिसतोय, ही सेल आहे का? नाही. नाव आय राईट द वर्ड दॅट इज this particular structure is showing the view inside the nucleus. Inside the nucleus. झालं, सिंपल आहे, काही नाही. आपण न्यूक्लियसच्या आतमध्ये बघतोय. दिस इज न्यूक्लियर मेम्ब्रेन अँड दिस इज न्यूक्लिओलस. प्रॉमिनंट आहे, क्रोमोझोम्सचे थ्रेड्स आहेत, ते क्रोमॅटिन फायबर्स होतं. आता मिओसिस होणार आहे, सगळं रेडी होणार आता असतं. ते रेडी होणार म्हटल्यानंतर त्याला काहीतरी थोडीफार केमिकल प्रोसेस आहे, थोडं अट्रॅक्शन आहे, कॉम्पॅक्शन आहे, सगळं होतं. आपल्याला काय करायचं आहे, आपल्याला फक्त आज लेप्टोटीन बघायचं आता. ठीक आहे. सो दिस इज लेप्टोटीन. डबल थ्रेड chromosomes, they have appeared and now they are showing their nature thin and they are showing their presence prominently, distinctly they can be seen. So this is leptotype. Once again, let me remind you one more thing. We are talking about the sex cells. We are not talking about the somatic cells, body cells. We are talking about the sex cells. This is the actual sequence. Leptotyne appears there in sex cells only. मी लेप्टोटीन जर का म्हणलं की स्किनच्या सेलमध्ये लेप्टोटीन फेस बघितली, तुम्ही म्हणाल अरे हा काही सांगतो, असं काही लेप्टोटीन दिसते का स्किनच्या सेल्समध्ये? नाही दिसणार. स्किनच्या सेल्समध्ये मायटोसिसच दिसेल, इथेच मिओसिस दिसेल. हेच आपल्याला लक्षात घ्यायचंय. हे ज्यांना आज कळालं, निम्मा चॅप्टर तुमचा झाला तयार. आलं लक्षात? इतकं सोपं असतं. हे सगळे इव्हेंट्स आहेत. कुठलेही या इव्हेंट्समध्ये या पेज नंबर 24 वरती अवघड काही नाही आहेत. निवांत राहा, शांत राहा, सगळं होणार आहे, सगळ्यांना सगळं जमणार आहे. आणि शाळेपर्यंत म्हणजे 10वीपर्यंत तुमच्या स्कूलमध्ये इफ यू हॅव नॉट डन एनिथिंग, लिटरली इफ यू हॅव नॉट डन एनिथिंग विथ दिस पर्टिक्युलर नॉलेज, डझंट मॅटर. आता तुमची सुरुवात होते, हरकत नाही. जी कधी सुरुवात झाली ती चांगली झाली असं समजायचं. सो दिस इज लेप्टोटीन. चला याच्यातलं एक वाक्य वाचून टाकू आपण. whatever the things are there in the leptotyne part, let us read the things together. Chromosomes become distinct. Yes, chromosomes they become distinct. Now they are in the form of thin threads. Still there is no necessity of getting the things compacted. Thin rahu de na. Why to waste energy? Let us save the energy. Let there be the chromosomes in the proper form, but let us get ready for the actual division phase. So the chromosomes they become alert, that is distinct. Second, they are in the form of thin threads. Since there is no compaction, the chromosomes will appear in thin threads. Third one, chromosomes appear to be single, but actually each one it has double threads. So we are talking about sex cells. We are talking about this particular nucleus. We are on nucleus मध्ये झाकून बघतो here. So we are talking about nucleus, and if you think about this particular patch, and आता हे जर का खूप लांबून बघितलं, simply it will appear like this. Hence that statement is given, chromosomes appear to be single, but no, they are not single. If you observe the things carefully, not actually, but carefully, then the chromosomes will show two threads which are bound together. Okay? आता इथे तुम्हाला हे सगळं माहिती आहे. हा इथे सेंट्रोमियर आहे. ओके. नाउ न्यूक्लियर मेम्ब्रेन अँड न्यूक्लिओलस इज प्रेझेंट. न्यूक्लियर मेम्ब्रेन कुठे सांगा बरं? हे इथे न्यूक्लियर मेम्ब्रेन आहे. सगळं आतमध्ये बांधलं गेलेलं आहे. What about nucleolus? Nucleolus is present. For convenience, I have used a common title for both the names, but whatever the form is available for a specific nucleus, it appears to be different. Remember. So this is nucleus, this is nuclear membrane. So these are the chromosomes. Okay. This is the first stage of प्रोफेज 1 झालं. किती स्टेजेस होते आपल्या? 5. जे प्रोफेज 1 च्या झाली. पहिली शिकलात तुम्ही. काय होतं त्याच्यात? काय नाही. क्रोमोझोम्स, they are distinct. They appears to be thin. Whenever you look at the chromosome, it looks like a single thread, but actually 2 threads are there. झालं, संपलं. तुम्हाला जमलं. कारण आता पुढे टेट्राड किंवा बायव्हॅलेंट हा जो कन्सेप्ट आहे त्यावेळेस तुम्हाला लक्षात ठेवायचं. I am going to remind you about this particular terminology. Okay. Now let us move to the second diagram, or I can say phase B or stage 2. It is popularly called as zygote. ठीक है, अपन ये zygote मान लेना. So this is zygote. Yes, there are certain there is a new word, synapsis, not synopsis. It is a synapsis. Chromosomes, they contract, they become compact, they contract lengthwise, become shorter and thicker. If they are contracting lengthwise, definitely they will be shorter as compared to the previous one. And they become thicker. बरोबर आहे. लेंथवाइज होतं, कॉम्पॅक्ट केलं, sideways it will grow, it will show some increasement. So definitely they will become shorter and thicker. Okay, agreed? Now here we are talking about the most important term, that is homologous chromosomes. Whenever we think about homologous chromosomes, in that particular case the crossing over is possible. In case of heterologous with different type of chromosomes, the exchange cannot take place because whatever the genes are required for crossing over, they are not placed on the same loci, on the same position. Forget about the word locus. They are not present on the same location. Okay, here we are considering the homologous type of chromosomes. So homologous type of chromosomes, they come together, they form a pair. That pairing is called as synapsis. It's a simple pairing, clubbing. आपण कसे असतो ना की स्कूलमध्ये सांगतात, ग्रुप असेल, एक मोठा सर्कल असतो आणि त्यामध्ये सांगतात की ते जसे ऑर्डर्स देतील त्याप्रमाणे आपल्याला 2-2ची जोडी तयार करायची असते, पेअर तयार करायची असते. त्या टाइम टीचर और एनी स्पोर्ट्स इंस्ट्रक्टर he says that now you need to make a pair with same color. If I am wearing this particular light green color, then I will go for searching the light green color. If someone is wearing blue color, he will go for in search of blue color. So this is how the pairing will occur. Similarly, this homologous chromosomes, they will come together. They will form a pair. There will be a synapsis. There will be a pairing. So this is how the pairing. These are the chromatids. They are getting close to each other because this particular homologous chromosome, they are getting together. Okay, as I mentioned in the beginning, there are total 23 pairs, means 46 chromosomes. 22 plus 1. This particular number is present with the human cells. Okay, now let me tell you about this particular diagram. Instead of 46, simply we have shown 4 to the 8 number of chromosomes with double chromatids. Okay, so this is how the two homologous chromosomes, they get paired with each other. Okay, with similar properties, similar genes, similar shape, similar size, dimensions, they get into the pair. Okay, this is the basic concept. Let us read about zygote, this second stage from your notes. Chromosomes, they contract and become shorter and thicker. Yes, we have learned this. Homologous chromosomes come together and they form a pair. This is known as synapsis. Synaptonemal complex is developed, and let me tell you, this is the particular word. So is it legible, sir? Yeah. So this synaptonemal complex is formed here. Where this particular synaptonemal complex will be formed? At the point of synapsis. Wherever the chromosome, homologous chromosome will form a pair, at that particular point there will be a synaptonemal complex will get developed. So first synapsis will occur in zygote and synaptonemal complex will get formed in the zygote. Okay, now this is all about the second phase. Are you getting the concept? Do you find it difficult? If you are finding this particular concept difficult, then simply pause the video, go back and listen to the concept, understand the concept once again, refer to the notes, read your NCERT book. As far as possible for this particular chapter, do not read your board book much. There is one more important concept I'm going to tell you. Let us go through the things first, and I'm going to repeat all these things in the next few minutes. Okay, let us be here with the stage 3. This is patching. Patching आहे, त्याच्यामध्ये काहीतरी आता पुढे व्हायला पाहिजे, बरोबर आहे. कारण आता सगळं झालं, तुम्ही एकत्र यायला सांगितलं, आम्ही आलो, पेअरिंग करायला सांगितली, केली. आता पुढे जी ॲक्च्युअल प्रोसेस आहे की ज्याच्यामध्ये क्रॉसिंग ओव्हर हा शब्द ऐकला ना की आपल्याला वाटतं यार काहीतरी भरपूर याच्यामध्ये टेक्निकल पार्ट जास्त आहे, सोडून देऊ, हा पार्ट नंतर करू. तसं वाटत असेल तर तसं करा, पण हा कन्सेप्ट सोडून दिला की उद्या तुम्हाला जेनेटिक्स, बायोटेक्नॉलॉजी आणि चुकून तुम्ही ह्युमन जेनेटिक्स घेऊन काही मेडिकल नंतर स्पेशलायझेशन करणार असाल, हे लागणारच आहे ना सगळं. खूप कमी लोक मेडिकल नंतर रिसर्च सेक्टरमध्ये जातात आणि त्यामुळे थोडं आपला कुठेतरी नॅशनल लेवलला आणि ऑब्व्हियसली इंटरनॅशनल लेवलला थोडा रिसर्चमध्ये इम्पॅक्ट कमी पडतो. ठीक आहे, खूप लोक तुमच्यातले म्हणतात बरं का, आम्हाला आयसरला जायचं आहे, आम्हाला KVPY करायचं आहे, तुम्ही IISCची माहिती द्याल का, कोणी NCBSला आमचे सिनियर्स आहेत का. अरे सगळीकडे सगळी लोक आहेत. सिम्पली नॉक अँड इट विल ओपन, हे कायम लक्षात ठेवा. पण त्याच्याकरता आधी बेस तयार करायला पाहिजे. ते नॉक करण्याकरता आधी बेस लागतो आपला, येते लक्षात. सो दिस इज द टिपिकल थर्ड स्टेज, दॅट इज पॅचिटिंग. इथूनच आपण आता वाचूया. इथे तुम्हाला काय काय दिसत आहे. सो दीज आर दी सिस्टर क्रोमॅटिड्स. सिस्टर कोणाचे आहेत पण, सपोज दिस इज फॉर युअर कन्व्हिनियन्स आय एम सिम्पली making this part clear just to put some label over here. Suppose this is chromosome 1 and this is chromosome 2, C1 and C2. आता क्रोमोझोम 1, 2 मध्ये सुद्धा हे जे दोन आर्म्स असतील, क्रोमॅटिड्स असतील. क्रोमोझोम 1 चा हा आर्म 1, आर्म 2. क्रोमोझोम 2 चा हा आर्म 1, आर्म 2. So these are the sister chromatids. Yellow, they come together. Blue and blue. So these are the sister chromatids. So always remember, whenever the crossing over occurs, it occurs with non-sister chromatids. इथे वर्ड आहे बघा. आपल्याकडे सगळ्या गोष्टी तयार आहेत. आपण सगळ्या गोष्टी व्यवस्थित वाचलेल्या पाहिजेत. आपल्याला नॉलेज सगळीकडून मिळत असतं, ते योग्य वेळी घेता यायला पाहिजे. सो पॅचिटिंग. तर मी इथे वाचतो, डायग्राम पण बघू. Chromosomes become still shorter. Yes, now there should be proper condensation, there should be proper compaction, because now there will be the vital process of exchange. Okay. Each chromosome consists of two sister chromatids. This is chromosome 1, chromosome 2, and there are two chromatids for each chromosome. So if you consider this as a pair, total chromatids will be 4. 1, 2, 3, 4. Okay. Therefore, each bivalent consists of 4 chromatids. हे bivalent काय concept आहे हा प्रश्न पडतोच. So this is bivalent. When I talk about pairing, हे दोन लोक एकत्र आलेत, हे दोन क्रोमोझोम्स एकत्र आले, प्रत्येकाकडे दोन आर्म्स आहेत. दिस इज आर्म 1 2 1 2. आपल्या कन्व्हिनियन्स साठी त्याला 1 2 3 4 केलंय, पण टेट्राड म्हणायचं आहे ना आपल्याला, त्यासाठी दिस पर्टिक्युलर सेट इज कॉल्ड ॲज bivalent. येथे concept लक्षात. Once again, when we talk about the zygotine phase, in this particular phase the homologous chromosomes, they come together and form a pair. Now the pair is formed. Here also some more details are available, but let us think about the actual tetrad and bivalent compound concept. This particular arm 1 2 3 4, when they are present together, it is called as, it's a tetrad. When you consider this particular set as a one set, you can call it as bivalent. Okay. या कन्सेप्ट तुम्हाला लक्षात ठेवावे लागतील आणि हळूहळू त्या वापरूनच येतात लक्षात. Patching. Crossing over takes place between non-sister chromatids. So always remember, the crossing over always takes place with non-sister chromatids only. 1 2 मध्ये होईल का? नाही. 3 4 मध्ये होईल का? नाही. Recombination or crossing over is possible with 1 and 3, 1 and 4, 2 and 3, 2 and 4. So this is how the things are possible. Fifth point. During this process, segments of chromatids are exchanged by breakage and fusion. आता कटिंग जॉइनिंग हा शब्दही सोपा आहे की नाही. चला ब्रेकेज आणि फ्युजन पाहिजे आपल्याला. कटिंग झालं, ठीक आहे, एक पीस कट झाला, दुसरीकडे दुसरा एक पीस कट झाला, मग नॉन सिस्टर होते, एकाने हा एक पीस काढून त्याला दिला आणि त्याचा काढलेला पीस आपल्याकडे घेतला. Now the actual proper exchange has occurred, and this is how the crossing over is getting successful. It is not completed. The status is not complete. It is in the process, but still you can say there is breaking and fusion, that is cutting and joining is taking place. There are certain enzymes which are required. Obviously, endonucleases, they are always used for breaking, and ligases, that is ligation, joining, so that is for joining, that is annealing. Annealing शब्द छान आहे. Annealing तुम्हाला लक्षात राहिला तर मस्त आहे. Once again, there are certain enzymes which are required to cut the specific piece of DNA, that is gene, and it should be placed with another chromosome, and different piece which is showing the same property or which is having the same locus, so that piece should be placed with the previous one chromosome. त्याच्याकरता ते cutting chemical scissors, they are required. You should cut it, put it somewhere else. Another piece you should cut it, get it here, then paste it, and the previously cut part you should paste it there. इकडचे तिकडे तिकडचे इकडे करायचे. So for that, cutting type of enzymes are required and joining type of enzymes are required. Point number 6 बघा परत एकदा. Point number 5. Endonucleases, they are used for breaking, that is cutting, and ligation, ligases, they are used for joining. You can call it as annealing. शब्द लक्षात राहिला बेस्ट, नाही राहिला अजून त्याहून सुद्धा जॉईनिंग लक्षात ठेवा, फक्त लायगेशन लक्षात ठेवा. लायगेशनला तुम्हाला सोय नाही आहे. लायगेशन दॅट इज जॉईनिंग. ओके. The last one point is in this way the genes are exchanged, the genetic recombination occurs. So is it visible here? Okay. Genetic recombination occurs here with the patching. त्याच्यामध्ये आपण न समजण्यासारखं काय ते आता बघूया. Actually, some of you may get confused with this particular tetrad and bivalent. Some of you may get confused with crossing over and synapses. Some of you may feel that what is this synaptonemal complex and what is this crossing over, how they are related to each other. Let me tell you in a very simple manner. There are so many technical knowledge available for this particular phase. Even if you search with this particular terminology, zygote or patching, with good reference books, entire chapter is given for the same. Okay, so when the homologous chromosomes they come together, they form a pair. This pairing is called as synapsis. When the synapsis occurs, joining occurs, that time there is a formation of synaptonemal complex. So this is how the synaptonemal complex continues to be there in the next phase, that is of patching. That is third phase. In patching, the actual crossing over occurs. If you do not remember, if you are not able to remember the word crossing over, you can simply say exchange of DNA material takes place. Crossing over occurs between two non-sister chromatids. When two non-sister chromatids are exchanging DNA material, that time some piece of gene, piece of chemicals, they are getting exchanged. Okay. Now, I think we have done with this board, and we need to shift the camera to the third board with a small pause. Now, let us think about this particular board. We have done with 3 actual steps. Actually, थोडं वाटू शकतं की ह्या 5 स्टेप्स आहेत आणि ह्या कशा आहेत, काय आहेत, आपल्याला कदाचित कन्फ्युजन वाटू शकतं, पण तसं काही नाही आहे, सोपे आहेत. ऑलरेडी वी हॅव डन विथ पॅचेटिंग टाइप ऑफ थिंग. ओके, अंडरस्टूड. क्रॉसिंग ओव्हर हॅज अकर्ड, DNA material has been exchanged, that is chromatid exchange has occurred, and now genetic recombination has occurred. Let us think about the diplotin. The chiasmata. Chiasma formation or chiasmata formation is another term. Synapsis, crossing over, and genetic recombination we have learned. Now think about the this particular fourth phase, that is diplotin. What is diplotin? Diplotin is nothing but the dissociation of synaptonemal complex. What do you mean by dissociation? Initially, there was no requirement of association of actual homologous chromosomes. Homologous chromosomes, they come together, they form a pair, then crossing over occurs, and once the crossing over has occurred, now there is a necessity of getting the things separated. So compaction होतं, chromosomes were in compact form. Now you need to understand there is a necessity of for things to get separated, because this particular cell need to enter into the last phase and then to the second phase of meiosis. Okay, now let us read about the features of this particular diplotin. Diplotin, that is dissociation of synaptonemal complex. So here the synaptonemal complex was formed, and in that particular complex the crossing over has occurred, and now that particular part should get dissociated. वेगळं व्हायला पाहिजे, अलग होना चाहिए. So this particular thing happens there. Centromere moves away. This particular thing you should remember. The centromere, they move away. Centromere move away, and definitely this particular diplotin type of thing is ready to enter into the last phase of the prophase 1. Still we are here in prophase 1. We are here in prophase 1. मी सतत सांगतोय, तुम्हाला लक्षात येऊ दे, आत्ताशी मिओसिसच्या ज्या टोटल 8 स्टेजेस आहेत त्यातली पहिली स्टेज चालू आहे. पहिल्या स्टेज मधली ही फेज जी आहे ती 4th फेज आहे, अजून 5th फेज व्हायची आहे. ओके, सो दिस इज diplotin, that is dissociation of synaptonemal complex occurs, and then centromere they move away. Centromere मगाशी जवळ आले होते सगळे, ते आता इथे जवळ आलेले दिसतील, जवळ आलेले दिसतील, इथे जवळ आहेत, आता हे लांब जात आहेत. सो दे आर गेटिंग अवे फ्रॉम ईच अदर. दिस पर्टिक्युलर पॉइंट, आता ही हिस्ट्री झाली. दिस पर्टिक्युलर पॉइंट ऑफ चायझमॅटा फॉर्मेशन, और इथे पेरिंग झालं होतं, जिथे क्रॉसिंग ओव्हर झालं, जिथे एक्सचेंज झालं. सो दॅट पार्ट इज हियर which is represented with this particular color. Now this particular point is called as point of chiasma formation. Now look at this particular nuclear membrane. So slowly the things are getting loosened. आता हे membrane जाणार आहे, envelope is going to get dissolved in one sense you can say. Let us enter into the last phase. So this last phase is all about the shift of chiasmata towards end, and nucleolus, nuclear membrane disappears. तो इथे हळूहळू thin होत जात आहे. Deliberately I have shown this particular nuclear membrane with thick line, here thin line, and as this particular part ends, that time the nucleolus and nuclear membrane will disappear completely. आता इथे मी तर दाखवताना हा न्यूक्लिओलस दाखवलाच नाहीये, इथे थोडं थोडं कमी होत चाललेलं आहे, त्याची डेन्सिटी कमी होत चाललेली आहे. डायकायनेसिस, कायस्माटा शिफ्ट टू द एंड. हा बघा इथे जो काही अटॅचमेंट पॉइंट होता, सो दॅट पॉइंट इज गेटिंग शिफ्टेड टू द एंड. न्यूक्लिओलस अँड न्यूक्लियर मेम्ब्रेन इट डिसअपियर्स. आता अटॅचमेंट जर का अशी झालेली असेल ऑलरेडी, असं सेपरेट झालं, ओनली दिस पर्टिक्युलर एंड पार्ट will remain in contact with each other. So this is how chiasmata initially it was here. There are so many points. There are so many points are possible to be there for chiasmata on a single chromosome. But at the end of prophase 1, in case of meiosis 1, the chiasmata shifts to the end, and this is how the actually chromosomes appear there in the nucleus. Nucleus means the nuclear membrane is slowly it is getting disintegrated. It has started disappearing. Nucleolus has already disappeared. Now this particular concept is called as shifting of chiasmata to the end is called as terminalization of chiasma. लक्षात येते आपल्या? छान वाटतंय? आता आपण परत पहिल्यापासून सुरू करूया. ही जी फेज आहे, दिस पर्टिक्युलर फेज वॉज द लास्ट फेज. In very simple terms, in very simple language, I have tried to explain this. Now we are going to I'm going to explain this particular concept again, but this time I am going to use little technical words more, and you need to understand the things better. Okay, let us shift to the first board. So let us look at the various stages of meiosis 1. This is leptotin, zygote, pachytene, diplotin, dikinesis. We have started with the leptotin. very initial phase, the sex cells which take part in actual cell division or formation of gametes, they always show meiosis. This is meiosis 1 we are learning, and specifically in meiosis 1 we are learning 5 different stages of prophase 1. So this is leptogene, the chromosomes which are present here in the nucleus. So they become distinct, they become prominent, though they appear thin by nature. Always remember, whenever we think about a typical leptogene phase, nuclear membrane appears to be prominent, nucleolus appears to be very prominent. Now, leptogene is a phase where the chromosomes are getting arranged, they are getting organized, just to show the further process, for the development. This particular thread-like thing is chromosome, and in normal cases one may find this particular structure as a single thread, but always remember there are 2 chromatids for each chromosome. There are 2 chromatids for each chromosome. We are talking about meiosis. We should consider this cell to be diploid. I am repeating the word. We need to consider this cell to be diploid. Okay, 2n. Status is 2n. Okay. Now, another concept, important concept of Bokeh stage. This particular Bokeh word, it is there with your board book. Okay, I think board book you can go through it, and on page number 79, leptogene, Bokeh stage, the word you can easily refer. Now let me tell you about the actual Bokeh stage. Whenever we think about any bouquet, how it appears, so I can show it here simply. काही जणांचा basic problem असतो, they cannot understand what exactly the word means. For them, I am showing this particular basic diagram. माझा सुद्धा हाच problem होता. So this is bouquet. Let us think about the cell now. Sorry, my mistake, nucleus. So this is the nucleus, the miniature of this. This is nuclear envelope. What I have drawn just now is nuclear envelope. So this spherical structure, somewhere here, say if this centromere is present and this chromosome is showing presence with centromere, the arms, and this is how, if the what I can say, I cannot say design, but if the position of chromosome appears like this, then this particular stage is called as Bokeh stage. Always remember the ends of the chromatids, they fix themselves to the inner side of the nuclear membrane. Let me tell you, suppose this is the nuclear membrane, inner side of the nuclear membrane, and suppose this is the chromosome, these are the chromatids. This particular notch part can be called as centromere. Now, this is how things will get attached. Another chromosome like this, another chromosome, another chromosome, another chromosome. This is how there will be a cluster type of thing which is getting attached to the inner surface, inner lining of the nucleus or nuclear envelope. This is how you can say this stage is Bokeh stage. तिथे तुम्ही असे विचार करू शकता. You can simply, you need to imagine, you need not to mug up the things. How things can appear? Yes, beautifully. This is simply a diagrammatic representation. slowly all of them, they will try to get attached to the inner surface. Both the ends will try to attach to the inner surface of the nuclear envelope. This stage is called as Bokeh stage. I'll use another color. So this is Bokeh stage. Let us talk about the zygote. I hope you are referring your diagram sheet. Everything is given there in a proper form. For our convenience, I have used blue and yellow color on the board, and on the diagram sheet number 2 on Meiosis 1, total 8 stages are shown. Diplotin, zygote, pachytene, diplotin, dikinesis, all the 5 stages are shown, and simply black and white coloration is actually possible. It was possible to use. Details of the prophase are given here with the diagram number 10. This particular diagram number 10 you can easily observe. Stage A, B, C, D, E, total 5 stages. Okay. Let us think about the zygote once again. Simultaneously, you are supposed to refer your diagram sheet. Zygote is a typical process where the homologous chromosomes, they come together. The chromosomes, they start contracting. When they start contracting, they become the space become more available for their movement and recombinations. Now the chromosomes, they have become shorter and thicker. Space is available. Homologous chromosomes are there, and they start forming a pair. When they start forming a pair, the synapsis occurs. Synapsis is nothing but the pairing. Pairing has occurred between the homologous chromosomes, and you are well aware about the concept of homologous. Homologous means the chromosomes which have a similar set of genes on a similar position or a similar loci with same dimensions of a chromosome. So this is homologous concept. When there is a formation of synapsis, when there is a pairing, there will be a synaptonemal complex formation. Now this particular stage is going to enter into the next stage. Cell is going to enter into the next stage, that is pachytene. That time you need to understand what is synaptonemal complex. Synaptonemal complex will be useful for actual recombination, exchange of DNA material, exchange of chromatid pieces. म्हणजेच काय, आपण DNA एक्सचेंज करतो, म्हणजे त्यातले काय केमिकल्स घेतोय, a specific set of gene. This is how we are turning to the next phase, that is pachytene. Now pachytene crossing over between non-sister chromatids has occurred, or you can say this particular crossing over will take place in the pachytene. Okay. Now, crossing over between non-sister chromatids, this particular concept is more important. Why non-sister? Because this particular 1 and 2, they cannot exchange the genes with each other. दोन्हीकडे सेम सेट असतो जीन्सचा, तो कसा एक्सचेंज होईल? To exchange something with each other, there should be some difference, there should be some requirement, there should be some reason to exchange. Though the gene locations for both the homologous chromosomes are same, even then slight variations are possible. साधी सरळ गोष्ट आहे, दोन भाऊ असतात, त्यांच्यात 1-2 वर्षाचं अंतर असेल, पेरेंट्स आहेत तेच आहेत, त्याचे जीन्स तेच आहेत, पुन्हा क्रॉसिंग ओव्हर होत आहे, कॉम्बिनेशन किती होऊ शकतात. दॅट पर्टिक्युलर पार्ट वी आर गोइंग टू लर्न how many complex combinations are possible, how variation is going to take place. We are going to learn that, but before that you need to understand why this particular process is happening. Okay, so crossing over between non-sister chromatids, it takes place. The exchange between chromatids means there is an exchange between DNA molecules. Chromosomes may uncoil केला कि what will happen? It will show a continuous long thread of DNA molecule. Now some pieces of DNA are getting exchanged with non-sister chromatids with some another homologous chromosome. Always remember, this is how the genetic recombination has occurred for the first time. Let me ask you a question. Genetic recombination is occurring here at this particular phase. Understood? Chapter is over. Now tell me how 4 different haploid cells with totally different genetic recombination is possible. If genetic recombination is taking place here in this particular Meiosis 1, Prophase 1, specifically in this particular Pachytene phase, then how is it possible to have 4 different genetic combinations with a haploid cell as a result of meiosis? Here we are thinking about a single cell only. Actually, it happens with each and every sex cells. Okay, now मला एक सांगा की एका सेल मध्ये क्रॉसिंग ओव्हर झालं, ती परत सेपरेट झाली, 2 कॉम्बिनेशन्स तयार झाली, हे मी मान्य करतो. Then how is it possible to have 4 different haploid cells with 4 different totally different genetic recombinations? Think about it. Definitely I am going to explain about this, but before that you should think about it. Okay, now crossing over has occurred, genetic recombination has taken place. Now it is time for a cell to enter this particular nucleus into the another phase, that is diplotin. So can it be seen properly? Okay, diplotin, that is dissociation of synaptonemal complex. Earlier it was an association, pairing, joining, annealing was there. Okay, now there is dissociation. The process is completed. Why to waste energy keeping the material in a compact form? Okay, let us make it separate. So this is how the diplotin phase is specifically known for dissociation of synaptonemal complex. And centromere, they move away. The centromere, they move away. हे तुम्ही anaphase मध्ये वगैरे तुम्ही ऐकलेलं आहे आता. ते जायचंच आहे आपल्याला. पण ह्या सगळ्या गोष्टी, हे सगळे इव्हेंट्स हे लक्षात यायला हवेत. त्याच्याशिवाय तुम्हाला कॉन्फिडन्स वाटणार नाही की मला मला हे जमू शकतं. त्याच्यासाठी इव्हेंट्स लक्षात घ्या. सेंट्रोमियर, द पॉइंट, इथे जंक्शन पॉइंट जो असेल, सो सेंट्रोमियर्स दे मूव अवे. A centromere cell, suppose, so it moves away. So this is how the things are taking place. Centromere moves away, and now the dikinesis occurs. Terminalization, तो word आपण आता लक्षात ठेवला पाहिजे. Okay, dikinesis, chiasmata, that is the bearing, the point of chiasmata, you can see here. So it moves to the end. It moves to the end of the chromosomes. So this is how the things appear now. So this is terminalization of chiasma. This particular phase is called as terminalization of chiasma, that is dikinesis. Nucleolus and nuclear membrane, it disappears. या सगळ्या फेजेस तुमच्या आता लक्षात आल्या. प्रोफेज, मेटाफेज, एनाफेज, टेलोफेज तेच आहे. फक्त याच्यामध्ये काय झालं, प्रोफेज 1 आली, तसंच प्रोफेज 2 पण आली, तसंच मेटाफेज, एनाफेज 1, 2, टेलोफेज 1, 2 या सगळ्या आल्या. प्रोफेज 1 ला थोडासा जास्त त्रास आहे. इलेक्ट्रोटीन, झायगोटीन, पॅचिटीन, डिप्लोटीन, डायकायनेसिस, 5 स्टेजेस शिकाव्याच लागणार. हे एकदा आलं लक्षात की आता आपण पुढच्या टप्प्यात जाऊ शकतो. नाउ दिस पर्टिक्युलर पार्ट इज एंड ऑफ Prophase 1. We are going to enter into the Metaphase. Metaphase मध्ये बघूया काय काय होत आहे. बरोबर आहे. So I think we need to switch to the board. Okay. So we have done with the Prophase 1. Now further stages are Metaphase 1, Anaphase and Telophase 1. Now you need to understand already terminalization of chiasma has occurred. Refer your diagram sheet, page number sheet number 2 and diagram number 8 and a typical diagram number E from diagram number 8. Okay, already terminalization of chiasmata has occurred. Okay, now consider that particular sets of chromosomes. So I can draw a thing in a very crude manner. That is, suppose this is how the terminalization of chiasma has occurred. Now these chromosomes are going to get arranged on an equator in the middle, somewhere middle, and that phase is actually metaphase. And yes, you are well aware about the metaphase properties. You can read them from the notes or you can read them here also. Chromosomes, they come at equator at the typical middle of a available area. Okay, now spindle fiber formation, it starts. Centrioles are present. Now spindle fibers are formed. Now there should be a pulling mechanism, and yes, now the cell is entering into the anaphase 1. In anaphase 1, centromere, they do not divide. The double homologous chromosomes, they move away from each other. If you remember the actual diagram, then you can remember in this way. Already the terminalization of chiasmata has occurred. Now the things, suppose this particular pair is 1, the double homologous chromosome, this pair is 1. Now there is actually separation. Everything is arranged at equator. Centromeres are arranged at equator. Now spindle fibers are formed. There will be a pulling, and these particular two ends independently they are going to form two different cells. This particular reduction division has occurred here in meiosis 1. Okay. This is how the double homologous chromosomes, they are getting away, they are moving away from each other. This particular phenomenon of separation of chromosomes from chiasmata, इथे attachment होता ना सर, separation of chromosomes from chiasmata is called as disjunction. Once again, in the last phase, that is dikinesis, there is terminalization of chiasmata. Chiasmata have moved to the end, and now here in this particular metaphase 1, the chromosomes with the terminalization of chiasmata, so they start appearing on the equator. If they have started appearing on the equator, now you can say they should enter into the next phase, that is they should get separated from each other. Spindle fibers are already formed. The spinning action will start. Now you should remember centromeres are not dividing. Whatever the homologous chromosomes are there, so the double homologous chromosomes, if they are present like this, I am showing only two fingers, consider this as a double homologous structure, and these things are getting pulled away from each other. So this is how the reduction in chromosome number occurs. Other reduction division. Disjunction has occurred, that is separation of chromosomes from chiasmata. The joining होतं ते आता संपलं. Now they are separated just because of these spindle fibers. They have pulled them. Now there is reduction in chromosome numbers. झालं. आधी एकत्र आले होते, हे अशी पेअर होती की नाही, झालं तर झालं हॅप्लॉइड. आधी होतं हे डिप्लॉइड, आता झालं हॅप्लॉइड. येतंय लक्षात. Now this particular last phase of the meiosis 1 you should actually understand. So this particular stage ends with the karyokinesis. Here the karyokinesis, the nuclear division completes. So telophase 1, here the nuclear membrane and nucleolus reappear. In the last phase of the prophase 1, that is dikinesis, the nucleolus and nuclear membrane was totally disappeared. Now it has reappeared and the telophase 1 is about to complete. Once again the chromosomes become thin. Now there is no need of wasting energy in putting them in a compact form. So there can be actually the thinning of the chromosomes. Now let us think about the next phase. Keep your diagram sheet and the notes ready. Both the things there should be in your hand. Let us read about the meiosis 2 simultaneously. We are going to refer the diagram. Meiosis 2, that starts with the prophase 2. There is nothing new to say. It is just like ordinary mitosis. Meiosis 2 is nothing but the ordinary mitosis, or overall meiosis is highly modified mitosis. Once again, meiosis is nothing but the highly modified mitosis. Let us read about the meiosis 2. Prophase 1, the chromosomes are already doubled. They are thin and long. आता haploid set तयार झालेला आहे. Let us think about the meiosis 2. Diagram number 9, prophase 2, you can observe. Whatever the things were there in the telophase 1, the same thing is going to be there in prophase 2, that is first stage of meiosis 2. Now there will be the arrangement of chromosomes at the equator. Yes, duplicated chromosomes are going to get arranged on equator. Spindle fibers and spindle apparatus are produced as usual. If you are not recalling the actual stages of mitosis, then you should listen to the video. You should refer to the material once again, and then you will come to know the duplicated chromosomes. They are already arranged on the equator, and metaphase 2 has started. Spindle fibers and spindle apparatus are already formed and they are in action. Centromere of each chromosome divides. Look at the diagram number K. Diagram K of diagram number 9, that is Anaphase 2. The centromere of each chromosome, it divides. Then chromatids become separated and behave as a chromosomes. They now once again they are getting pulled away from each other, and now if they are showing the actual mitosis only, then definitely centromere divides and the chromatids, they behave as a chromosomes. Now the chromosomes start moving away from each other towards the respective folds. Same, the spinning mechanism has started, the continuous fibers and other type of fibers, means I can say anaphase movement you can recall. we have discussed in detail that anaphasic movement of chromosomes, because always remember the microfilaments, they are made up of all the microtubules. The spindle fibers are made up of microtubules, and microtubules, they always help in sliding movement of the any organelle. So here in case of actual cell division, the chromosomes and or the chromosome arms, you can say chromatids, are moving. So this is how the anaphasic movement once again it can be witnessed in this particular Anaphase 2. Now the cell is entering into the last phase of the meiosis, that is Telophase 2. Look at the diagram once again. Yes, I can say, yes, once again, the telophase is responsible for showing the 4 daughter cells which are haploid by nature. Chromosomes reach to the respective poles, then nuclear membrane is once again formed. Everything is applied for the newly formed cell. Now the karyokinesis is followed by the cytokinesis, that is division of cytoplasm. In case of plant cells, there is a plate formation. In case of regular animal cells, there will be no cell formation. There can be notch formation, then there can be ridge and furrow model. Everything is possible. So we are simply looking at the events. We are not going to enter into the details of each type of cell. Okay, simply we are talking about sex cells here, and we are talking about the general phenomenon. Now the karyokinesis is followed by cytokinesis, and now as a result of meiosis, 4 haploid daughter cells are produced. Let me tell you, this particular process of meiosis is a fantastic process. This particular process is of great importance. You can simply read the importance from the notes itself. Meiosis gives rise to haploid gametes because 46 chromosomes are of no use, so they should get reduced to 23. So 23 from mother's side, 23 from father's side, then that particular zygote will have 23 pairs, means 46 chromosomes. There are so many abnormalities, there are so many disorders with which these fortunate people, they survive. Actually, we should understand the importance of equal distribution of chromosomes. Even if a person gets 45 chromosomes, then also that particular person may show any type of defect. So we are not going to discuss that particular point here, but always remember this particular meiosis process is of great value. Let us have the second point. Due to meiosis, the diploid chromosome number is maintained. Due to meiosis, diploid chromosome number is maintained. Product is haploid daughter cells, but the statement is due to meiosis, the diploid chromosome number is maintained. This is because haploid gametes fuse to form diploid cell after fertilization. Egg cell and the sperm cell, when they come together, 23 plus 23, 46 means 23 pairs. Once again, they are going to give to the newly born baby or newly developing zygote. Okay, so this is how the things are there. The diploidy level is maintained. Crossing over during meiosis results in genetic variation, which is necessary for evolution. Evolution, once again, it's a great topic. I know it's a diffuse science, but evolution is a continuous and non-reversible process. Means initially there must be some mitosis process. We are not going to enter into the actual how the things have developed, but initially mitosis was there, and now there is a great necessity of meiosis. Hence the mother nature, it must have given a chance for organisms to produce the gametes, I can say, by meiosis process, where the actual recombination can occur. In mitosis, there won't be any recombination. In mitosis, there will be simply the addition. Here, the recombination occurs, crossing over. You can remember the word crossing over occurs, and this is how newly formed combination can be shared. In the beginning, a doubt was raised. In the beginning, a doubt was raised by me that if crossing over is occurring at one end only, then you will get two cells with two different combinations. Okay? Now, how you are going to get total 4 cells with 4 different genetic combinations? Now the answer is there with your diagram sheet. Look at the diagram sheet number 2, diagram number 8, a typical diagram for meiosis 1. Diagram number H, that is a diagram of telophase 1. Can you see that 2 daughter cells are formed? Okay. Now look at both the daughter cells, the upper side and lower side. Both of them, they have 2 sets of chromosomes, 2 plus 2, 4 total. Okay. Now this particular telophase 1, compare this telophase 1 with the prophase 2, that is the first phase of meiosis 2. Are you comparing? Okay. Now you can observe both the diagrams. You can observe both the diagrams. Diagram number H and I. Okay. Now, can you make out a difference between the chromosomes available? Look at the combination which is there with prophase 2. The upper cell, it shows some dark part at different place, and another lower part shows dark part with the chromosome at different place. Now these chromosomes, they are going to get pulled away from each other. The upper cell, intermediate product we can say, the upper cell, that is going to show regular mitosis, equational division. So equal division will be there, and 2 chromatids are going to get pulled away from each other. So this is how there will be one more combination. There will be one more combination available. The first crossing over has occurred and it has shown a great difference. The exchange has occurred. Now the cell is ready for the second step. In second step only, when the mitosis occurs, that time once again the things become, the haploidy level is maintained. This is how the chromosome numbers are getting reduced, and this is how the 4 different combinations of the genetic combinations of the cell can be seen. Now imagine about the huge number of combinations. Suppose in case of humans, if sperms are getting produced, and if it is getting produced by meiosis, then think about the millions of sperms are there, and at a time single cell produces 4, and you can divide the actual number, millions of sperms, by 4. Now think about number of cells and number of combinations. So that is just beyond imagination. Any calculator cannot work, any person cannot work. There are n number of combinations. Simply you can say, okay, say 1 million sperm count is present, then simply we can determine the actual recombination possibilities, probabilities. It is not like so. Let me tell you. I will clear this particular board. Now, let us talk about the not by blue, actually it should be, center here should be by white. Okay, now this particular patch is showing crossing over. There will be a formation of synaptonemal complex. There will be a genetic recombination. You can remember that word, or you can simply refer the images of board writing. So this is how this will be the point of recombination. Let me tell you, there can be a number of recombinations at these points, or at these points, or at these points. A number of recombinations are possible. You and me are just incapable of finding the number of permutation and combinations. The nature is great. This meiosis process is created by him, and in a single chromosome, in a single homologous pair of chromosomes, a number of combinations are possible. And think about the many cells with their chromosomes and their combinations, and how many things are going to be there. So every time always remember the simple mathematics, simple calculations, logic, the numbers will not always help you. You need to understand the concept. You need to understand the concept of meiosis. You need to understand the concept of crossing over, how diploid things are getting haploid and continuing into the haploid form. And when two haploid come together, they share their chromosomes and they become diploid. So always remember, there are a number of combinations available with meiosis, and meiosis is the only reason why the evolution is occurring, and evolution is the process just because of we are alive here and we are sharing the concept of meiosis. Okay. After this lecture, we are going to end up with a small session about revision of these chapters, these topics, mitosis and meiosis, through PowerPoint presentation. Then there is a small concept of amitosis that we are going to learn, and I think that part can be treated as a third part, or let us see how it can be clubbed together. But after this part, that small part will be there. Okay, till then you should revise the things, you should remember the concepts, you can practice these things in your practice notebook. Write everything. Always remember, there should be a notebook and pen with you. You should keep writing, you should keep jotting down the things, and then this particular teaching set will be of your use. Okay, till then, goodbye.